In the testicles of a male, a special process happens to reproduce gametes, specialized sex cells called spermatozoa. This process is called meiosis. Watch for these three significant events during the process as they are key events in achieving genetic variation: crossing over, independent assortment, and random fertilization. Crossing over. The chromosomes received from the mom's egg and the dad's sperm are not exact copies from their parents. They are recombinant versions of their chromosomes because of crossing over during prophase 1. Only one recombinant version is received from each parental unit, a sperm or an egg. Because of crossing over, the fruit fly will look genetically different from their parents. Independent assortment. With 8 chromosomes assorting independently in a fruit fly, each time independent assortment occurs, there are millions of possible assortments of chromosomes in meiosis in a fruit fly. Random fertilization. The ovum and sperm have millions of different possible chromosome combinations, and this number to the exponent 2 is the number of diploid combinations in every single zygote. All 3 of these events during the process of meiosis are key to attaining genetic variation. Today, we will be looking at meiosis in a male Drosophila melanogaster, also known as a fruit fly. The diploid number of a fruit fly is 8, the haploid number is 4, the gamete number is 4, and the chromosome number after meiosis is 16. Interphase 1, before DNA replication. The chromosome count starts as 2n equals 8. Interphase 1, during replication. During the S phase, the DNA and centrioles are replicated as shown in the image above. The DNA doubles to produce their respective sister chromatids, and 2 pairs of maternal and paternal chromosomes are formed during replication. This makes the chromosome count 4n equals 16. Interphase 1, after replication. As shown, pairs of maternal and paternal chromosomes become homologous pairs of centromere-bound sister chromatids. The maternal chromatids are non-sister chromatids to the paternal chromatids. This makes our chromosome count 2n equals 8. Prophase 1. As shown in the images, homologous chromosomes intertwine to form a tetrad through a process called synapsis. Then, crossing over occurs. This is when the homologous pairs trade genes with its counterpart. Genes from the maternal homologous pairs are given to the paternal homologous pairs, and vice versa. Watch as the non-sister chromatids exchange their genes. As mentioned earlier, this process develops recombinant versions of the chromosomes, increasing genetic variability in the homologous pairs. Metaphase 1. The nuclear membrane breaks down to allow the homologous pairs to move via the microtubules. The microtubules line the homologous chromosomes along either side of the metaphase plate. Independent assortment ensures that the side of the chromosome ends up on is entirely random, creating further genetic diversity. Because the homologous chromosomes line on either side of the metaphase plate, this ensures physical separation in 2 new daughter cells. With 8 chromosomes assorting independently in a fruit fly, each time this occurs, there are millions of possible assortments of chromosomes in meiosis in a fruit fly. Anaphase 1. Microtubules pull the homologous pairs to the poles of the cell, as shown in the images. The sister chromatids stay together when this happens. Homologous chromosomes have a combination of paternal and maternal genes that are now destined to be separated into 2 genetically different daughter cells. Telophase 1. As shown in the image, the nuclear membrane begins to reform and the cytoplasm begins to furrow. The chromosomes unwind into their chromatin form. Cytokinesis 1. As shown, the cell splits itself and 2 new genetically different daughter cells are formed. Now, n equals 4. Interphase 2. Centrioles are replicated. No DNA is replicated again. This only occurs in meiosis 1. Prophase 2. The nuclear envelope begins to break down and the chromosomes begin to condense. Metaphase 2. The recombinant chromosomes align themselves along the metaphase plate by the microtubules centromere centered. Each chromosome aligns randomly along the metaphase plate. This independent assortment of all 8 chromosomes yields millions of different possible results. The alignment ensures physical isolation into 4 haploid gametes. Anaphase 2. As shown in the image, the recombinant sister chromatids separate and move away from each other towards the cellular poles as microtubules begin to disassemble. The centrosome on each homologous pair dissolves as the sister chromatids separate. Now, 2n equals 8. Telophase 2. The cleavage furrow forms and the nuclear membrane reforms around the recombinant sister chromatids. Cytokinesis 2. The cells separate and 4 recombinant haploid cells result. In males, all 4 will become spermatozoa, and in females, only 1 will become an oocyte, while the other 3 become polar bodies. In the specific fruit fly, 4 will become spermatozoa. Now, n equals 4. After meiosis, DNA has duplicated and formed 4 new spermatozoa cells in the male Drosophila melanogaster.